Hi everyone, I'm Nicole and I will be doing my topic on proteomics. So, what is proteomics? Proteomics is the large scale study of proteins in an organism, system, or biological sample. It involves the identification, characterization, and quantification of all the proteins present in the sample. The experiment requires a positive and negative target. The proteomics experiment is essentially separating the proteins from the tissue, cells, or organism, then getting the protein structure information to identify the proteins and characterize them, and lastly, using a database to help read the results. The primary goals of proteomics is protein identification, that is determining the identity of proteins present in a sample. And one way this can be done is by mass spectrometry. Protein characterization, that is understanding the structure, function, and post-translational modifications of proteins. This involves studying aspects such as protein folding, interactions, and modifications like phosphorylation, glycosylation, and acetylation. There's protein quantification. This is measuring the abundance of proteins under different conditions or in different biological samples. Quantitative proteomics provides understanding into changes in protein expression levels in response to various stimuli or during disease. Protein-protein interactions. This is investigating how proteins interact with each other to form complexes and networks within cells. Understanding these interactions is crucial for making sense of cellular processes and functions. Some techniques that are used in proteomics are mass spectrometry, two-dimensional gel electrophoresis, which we talked about electrophoresis in class, liquid chromatography, and protein microassays. So there's some limitations and some advantages to this technique. Limitations first, the dynamic range. The dynamic range of a protein abundance in biological samples is vast, with some proteins being very high in abundance while others are present in really low amounts. Current proteomic techniques may struggle to accurately detect and quantify proteins that are in such a wide dynamic range. Sensitivity. Detecting low abundance proteins, especially those involved in signaling pathways or disease biomarkers, can be challenging. The sensitivity of current proteomic methods may not be sufficient to capture the entire proteome, particularly for proteins present in low concentrations. The identification of post-translational modifications. Proteins undergo various post-translational modifications such as phosphorylation, glycosylation, and acetylation, which play crucial roles in cellular regulation. Identifying and characterizing these modifications accurately is a complex task and often requires specialized techniques. Sample complexity. Biological samples, such as tissues or biofluids, are highly complex and heterogeneous. The presence of interfering substances, variations in sample preparation, and differences in sample types can affect the accuracy and reproducibility of proteomic analysis. And a big one is instrument costs. State-of-the-art mass spectrometry equipment and other proteomic technologies can be expensive to acquire and maintain. This can limit the accessibility of proteomic methods to certain research groups or institutions only. Some advantages. Although transcriptomics provides a useful overview of global gene expression, proteomics is often used as a complementary technique that provides a comprehensive insight to the protein profile of an organism. So, proteomics is more in depth than transcriptomics is. And through a proteomics approach, researchers can detect more protein targets than genomics, which leads to highly effective clinical trials and biomarker studies. These trials and studies result in novel drugs that better treat diseases. In this paper, colonic tissue was tested using antibody microarray analysis. The proteins gave a specific signature for colorectal cancer than other tumors do. The cells were put through a microarray analysis and then 24 different antibodies were added. The results are in this photo. The photo shows the same slide at two different wavelengths in A for Alexaflor 555 and Alexaflor 647. The red are tumors and the green are normal cells. In B, it's just an up-close shot of the wells showing the intensity of the fluorescence. In C, this is their reproducibility, showing the majority of the data points are standard with a few outliers. In this figure, this was from a paper that did analysis from multiple sclerosis. So this is proteomic analysis of multiple sclerosis lesions. Essentially what they did was they took fresh frozen multiple sclerosis brain samples, and then they cut them into slices for histological characterization, and then they did laser capture microdissection, which is what LCM is, to isolate the multiple sclerosis lesions. They did serial protein extraction on the SDS page. They did some mass spectrometry on it, and then they found the unique APs, CAPs, and CPs. In figure B, this is representative tandem mass spectra of peptides identified from tissue factor. So all of the peaks are from the mass spec, and it attributes to the underlined lines in the box. So all the peaks attribute to one of the underlying lines. On the left axis, you have relative abundance, and on the bottom axis, you have your underlined proteins. And then in C, you have your representative tandem mass spectra, but this is from your CAP samples. Same thing, your relative abundance is on the left, and then your underlying protein sequences are underneath. In this paper, the researchers developed a proteogest, which is a program that can generate basic statistics for intact and proteolytically processed proteosomes. In this paper, they demonstrated what 20 amino acids would look like in representative organisms. In figure 2a, they show their model organisms' names on the right and the length of the amino acids on the bottom. Proteome fraction is on the left. In figure 2b, they were able to program a chart for mean numbers of residues per protein. This breaks down the average amount of each amino acid found during proteomic experiments, making the data analysis a little easier on the researchers.